Good day, Sir Victor. We are Group 3, and this is our first laboratory discussion. In this laboratory discussion, we should be able to explain the different stages of the cell cycle that will be identified using a whitefish embryo at a blastula stage. Our objectives are: first is to identify the phases of a cell cycle. Second is to identify the structure associated with each phases of a cell cycle using the whitefish blastula as a specimen. And lastly, differentiate the different stages of mitosis.
With the provided materials, we were able to come up with results that revealed the different stages of mitosis. First up, we were able to find Anaphase. As you can see, each chromosome's sister chromatids separate and move to opposite poles of the cell. Upon separation, every chromatid becomes an independent chromosome. Meanwhile, changes in microtubule length provide the mechanism for chromosome movement. More specifically, in the first part of Anaphase, sometimes called Anaphase A, the kinetochore microtubules shorten and draw the chromosomes toward the spindle poles. Then, in the second part of Anaphase, sometimes called Anaphase B, the astral microtubules that are anchored to the cell membrane pull the poles further apart and the interpolar microtubules slide past each other, exerting an additional pull on the chromosome. The next stage we identified was Metaphase. In this stage, as prometaphase ends and metaphase, the chromosome align along the cell equator. At least 2 microtubules extend from each chromosome's kinetochore, with at least 1 microtubule attached to each pole. The tension within the cell is balanced at this stage, and the chromosome no longer moves back and forth. The spindle is also now complete, with 3 sets of spindle microtubules visible. Interpolar microtubules extend from the spindle pole across the equator, almost to the opposite spindle pole, and astral microtubules extend from the spindle pole to the cell membrane. Kinetochore microtubules attach the chromosomes to the spindle pole.
The third stage we were able to identify is telophase. In this stage, the chromosomes reach the cell poles during telophase, the mitotic spindle disassembles, and vesicles containing pieces of the original nuclear membrane gather around the 2 sets of chromosomes. The lamins at each end of the cell are then dephosphorylated by phosphatase. The creation of a new nuclear membrane around each set of chromosomes is the outcome of dephosphorylation. The fourth stage we identified is interphase. This is where you can find the nucleus, and these are considered dense, darkly stained bodies formed by several chromosomal materials that code for certain RNAs. Near the nucleus is the centrosome that contains centrioles. And lastly, we were able to identify prophase. Take note that prophase is the first step of mitosis, occurring after the G2 phase of interphase has ended. The chromosomes of the parent cell, which were duplicated during the S phase, condense and become thousands of times more compact during prophase than they were during interphase. When seen under a microscope, each duplicated chromosome consists of 2 identical sister chromatids connected at a place called centromere, resulting in X-shaped bodies. Cohesin and condensin are 2 binding proteins that catalyze the condensation process. Condensin generates rings that coil the chromosomes into highly compact shapes, whereas cohesin forms rings that hold the sister chromatids together.
With that being said, we're all able to identify the name and parts of the different stages of mitosis by reading and watching the said references.